we are going to learn about focal adhesions and hemidesmosomes anchoring cells to their substrate. It is much easier to study the interaction of cells with the bottom of culture dish than with an extracellular matrix inside an animal. Consequently, much of our knowledge of cell matrix interaction has been obtained from the study of cells adhering to various substrates in vitro. The stages in the attachment of a cultured cell to its substratum are shown in figure 11:16. At first, the cell has a rounded morphology, as is generally true of animal cells suspended in aqueous medium. Once the cell makes contact with the substratum, it sends out projections that form increasingly stable attachments. Over time, the cell flattens and spreads itself out on the substratum. When fibroblasts or epithelial cells spread onto the bottom of a culture dish, the lower surface of the cell is not pressed uniformly against the substratum. Instead, the cell is anchored to the surface of the dish only at scattered discrete sites called focal adhesions. Focal adhesions are dynamic structures that can be rapidly disassembled if the adherent cell is stimulated to move or enter mitosis. Focal adhesions are dynamic structures that can be rapidly disassembled if the adherent cell is stimulated to move or enter mitosis. The plasma membrane is a focal adhesion, contains large clusters of integrins. The cytoplasmic domains of the integrins are connected by various adapter proteins like talin, alpha-actinin, and vinculin to actin filaments of the cytoskeleton. Focal adhesions may act as a type of sensory structure, collecting information about the physical and chemical properties of the extracellular environment and transmitting that information to the cell interior, which may lead to changes in cell adhesion, proliferation, and survival. Focal adhesion also play a key role in cell locomotion, during which the integrins develop transient interactions with extracellular materials. Focal adhesions are capable of creating mechanical forces or responding to such forces. These properties might be expected from a structure that contains actin and myosin, two of the cell's major contractile proteins. A cultured fibroblast attached to a gelated surface that can be deformed by focal local forces. The original surface contained a uniform grid pattern that has been distorted by traction. Such as gripping or pulling forces generated by focal adhesions at the undersurface of the cell. Here is a fibroblast lying on a bed of fibronectin coated posts. The posts are flexible and capable of being moved independently of one another. It can be seen in this micrograph that the tip of the posts are being deflected as a result of pulling forces exerted by the cell. During the process of focal adhesion formation, talin undergoes a conformational change that exposes binding sites on talin's rod domain. The binding of vinculin to these exposed sites promote the assembly of additional actin filaments. The cytoplasmic domains of integrin are also associated with protein kinase such as FAK, focal adhesion kinase, and SRC. The attachment of the integrin to an extracellular ligand can activate this protein kinase and start a chain reaction that transmits signals throughout the cell. The association of myosin molecule with the actin filament can generate traction forces that are transmitted to sites of cell substrate attachment. Here we can see a picture which depicts collagen is there, fibronectin, beta subunit is attached with the collagen, alpha subunit is also there. Vinculin protein will be there, alpha actinin can be seen, a myosin will be there, actin filament can be there, then the paxillin, SRC along with the cytosolic region, then the focal adhesion kinase signal transmitted to the nucleus. Focal adhesion kinase is something which links the signals to nucleus. So fibronectin, beta subunit, alpha subunit, collagen, vinculin, alpha-actin, myosin, actin filament, talin, focal adhesion kinase, signals transmitted to the nucleus, SRC, cytosol, nucleus, DNA. Then, experimental demonstration of forces exerted by focal adhesions. In this approach, fibroblasts are plated on a deformable surface that contains a visible uniform grid pattern. Traction forces generated by focal adhesions can be monitored by observing deformations or arrowheads in the grid pattern of the substratum to which the cells are adhering. A generation of force can be correlated with the location of fluorescently labeled focal adhesions. Scanning electron micrograph of a smooth muscle cell sitting atop a bed of flexible elastomeric microposts whose tips has been coated with fibronectin. The attached cell is seen deflected the position of multiple posts. The degree of movement of a particular post reflects the magnitude of the reaction forces exerted by the cell on that post. Acting on the opposite direction, mechanical forces applied to the surface of cells that can be converted by focal adhesions into biochemical signals in the cytoplasm. This process of signal conversion is described as mechanotransduction, and the focal adhesion is acting as a mechanosensor by recognizing the physical properties of the environment. The ability of cells to respond to physical forces is important in many cellular behaviors and can be illustrated by a study in which cells were allowed to bind to beads that had been covered with a coating of fibronectin. When the membrane-bound beads were pulled by an optical tweezer, the mechanical stimulus was transmitted into the cell interior, where it generated a wave of SRC kinase activation. Mechanotransduction is thought to be mediated by conformational changes in some of the adapter proteins, such as talin, that are induced by stretching. Such conformational changes can expose important binding sites in those proteins that were previously hidden, allowing additional protein molecules to be recruited to the adhesion complex. Activation of this protein kinase can transmit signals throughout the cell, including the cell nucleus, where they can promote changes in gene expression. Activation of either FAK or SRC can dramatically alter cell behavior. The importance of the physical properties of a cell's environment in influencing cellular behavior is illustrated by a study in which mesenchymal stem cells derived from adult bone marrow were grown on substrates of varying elasticity or stiffness. When these mesenchymal stem cells were grown on a soft, pliable substratum, such as might be encountered by a cell within the developing brain, the mesenchymal stem cells differentiated into nerve cells. When grown on a substratum of greater stiffness, the same cells differentiated into muscle cells. Finally, when they grown on even stiffer substances such as those that might be home to cells growing in a skeletal tissue such as a cartilage or bone, the mesenchymal stem cells differentiated into osteoblasts which give rise to bone cells. Focal adhesions are most commonly seen in cells grown in vitro, although similar types of adhesion contacts are found in certain tissues such as muscle and tendon. Within the body, the tightest attachment between a cell and its extracellular matrix is seen at the basal surface of epithelial cells. Focal adhesions are most commonly seen in cells grown in vitro, Although similar types of adhesion contacts are found in certain tissues such as muscles and tendon. Within the body, the tightest attachment between a cell and its extracellular matrix is seen at the basal surface of epithelial cells, where the cells are anchored to the underlying basement membrane by a specialized adhesive structure called the hemidesmosome. Hemidesmosomes contain a dense plaque on the inner surface of the plasma membrane with filaments coursing outward into the cytoplasm. Unlike the filaments of focal adhesions, which consist of actin, the filaments of the hemidesmosomes are thicker and consist of the protein keratin. Keratin containing filaments of hemidesmosomes are classified as intermediate filaments, which serve primarily in a supportive function. The keratin filaments of the hemidesmosomes are linked to the extracellular matrix region by membrane spanning integrins including alpha 6 beta 4. Like their counterparts in focal adhesions, these integrins also transmit signals from the extracellular matrix that affect the shape and activities of the attached epithelial cells. The importance of hemidesmosomes is revealed by a rare disease, bullous pemphigoid, in which the individuals produce antibodies that bind to proteins, the bullous pemphigoid antigens present in these adhesive structures. Disease is caused by production of antibodies directed against one's own tissue. That is, autoantibodies are called autoimmune disorder and are responsible for a wide variety of conditions. In this case, the presence of autoantibodies causes the lower layer of the epidermis to lose attachment to the underlying basement membrane and thus to the underlying connective tissue layer of the dermis. The leakage of fluid into the space beneath the epidermis results in severe blistering of skin. A similar inherited blistering disease, epidermolysis bullosa. Bullosa is a word something related with the blistering. So epidermolysis bullosa can occur in patients with genetic alterations in any one of the number of hemidesmosomal proteins, including the alpha 6 or beta 4 integrin subunit, collagen 7 or laminin 5. Hemidesmosomes are differentiated sites at the basal surface of epithelial cells where the cells are attached to the underlying basement membrane. Electron micrograph of several hemidesmosomes showing the dense plaque on the inner surface of a plasma membrane and the intermediate filaments projecting into the cytoplasm. Schematic diagram showing the major components of a hemidesmosome connecting the epidermis to the underlying dermis. The alpha 6 beta 4 integrin molecule of the epidermal cells are linked to cytoplasmic intermediate filaments by a protein called plectin that is present in the dark staining plaque and to the basement membrane by anchoring filaments of a particular type of laminin. A second transmembrane protein, BP180, is also present in hemidesmosomes. The collagen fibers are part of the underlying dermis.